Hello, Eugene Aiello here again for the next in our series. This is the most exciting, I think, video since we started this series. The first few videos were all about getting an understanding how electromagnetic energy interacts with pigments and with cytochromes, the heme molecule, the porphyrin rings, the aromatic amino acids. Also, it was very important in that very beginning first video was to establish the whole idea that most of the conventional models that we've learned are pretty basic and flawed. They use irreducible complexity to make up for the fact that most of their theories are not sound. When you enter electromagnetic energy into a biologic model, you start to get a whole different world. Many other options open up at the quantum level of exactly how these different interactions are occurring. Conventional models use the basic structural shapes and fitting, lock and key mechanisms, and charge dynamics to explain things as simple as an enzyme catalyzing a reaction. The quantum levels where these things are actually occurring, electron transfers are vital to life. That is how systems function. So in this video, we're going to talk about something very important, how biologic proteins act as semiconductors. That is how all protein structures function. Different mechanics that are inherent in the protein based on their semiconductor nature is going to govern how things function. How does an enzyme catalyze a reaction? How does an actin myosin filament cross bridge make a contraction? How do neurons fire? All the different things that in conventional models we just kind of have learned all the steps and memorized the steps diligently, but how was this actually occurring? Such as a great example was that ATP synthase wheel in the first video. Discharging a proton spins a wheel and somehow magically ATP is created. You know, when you start to evaluate these theories, you start to sit there and go, is this just something where you get to the higher powers controlling how these things are acting? Which we're not going to get into that, but the mechanisms, the quantum mechanisms can all be understood when you understand how biologic proteins act as semiconductors. So every function is driven by inherent semiconductor nature within proteins. So the core, those side chains, when I was talking about tyrosine, that 6-carbon ring that was in tyrosine and how it had those pi electrons, that's what I mean by these inherent nature, that what's in that structure of that amino acid. So proteins are produced by chaining amino acids together in codes, and that code is the DNA. So DNA encodes the different segments of how all these amino acid residues are going to join. When they join, they're going to be in a core, meaning that protein's going to kink, like I showed you in the last video, those alpha helixes, how those protein chains are going to kink, and there's going to be a center core, and in that center core is going to be where these residues are, where all these different semiconductor properties of that protein are going to occur. Beta-pleated sheets, extremely complex. Conventional science really doesn't even have an explanation, nor does conventional science even have a theory on how electrons move through these beta-pleated sheets. So I'm going to begin explaining semiconductors by what we know of now, the simplest of semiconductors. We all look at electronic devices as these complex structures when really they are extremely simple semiconductors. The semiconductors that Mother Nature is producing within these amino acid cores are highly complex. They wouldn't even be able to scratch how to understand how these are working. So we're going to first start off talking about a basic silicone-based semiconductor, which is what we use in electronics now. So silicone by itself has 4 valence electrons. Valence electrons are going to be very important. We're going to keep bringing this up. So keep in mind, valence electrons are how interactions are occurring. Silicone by itself, since it has 4 valence electrons, can interlink with itself and form a lattice. And we all know that lattice as glass. Glass is a wonderful insulator because when this structure is formed, there's no free electrons. There's no pi electrons as we were talking about before, but there's no electrons to move. So if you put a voltage across this without electrons able to move through with that voltage flow, it won't happen. So no free electrons are in silicone. I want to point out right now, carbon also has 4 valence electrons. So as we get in and start talking about semiconductors, I want you to keep in mind carbon could replace silicone. So what is a semiconductor? A semiconductor is when you take silicone lattice and you dope it. How they dope it, I have no idea. But what they're doing is adding different elements into that glass matrix, and they're changing the structure of that matrix. So a common semiconductor we use we'll add a phosphorus to the silicone structure. Phosphorus has 5 valence electrons. So now this structure is going to have free electrons within it. That would be called the N-type material. They also will dope a second material, the P-type material, with boron. Boron has 3 valence electrons. So now this structure is actually going to be depleted in electrons. So both of these structures are conductors, meaning if you go to put a voltage through that, electron flow can happen in these because there's free electrons to move. So how do we get a semiconductor out of this? When you join these two structures together, at that very edge, those free electrons are going to bump into places where there's no electron. We call these holes. So in the P material, you have holes, and in the N material, you'll have extra electrons. So right at the edge of this, the PN junction they call it, you're going to develop something called a depletion zone, where all those holes are filled up with electrons, and now those electrons don't have the capacity, the energy to jump. This is now an insulator. Electrical flow cannot happen if we put a charge across it. So if we do put a charge across it, which is what we do with electronic devices, that's how we're using semiconductors. If we hook a battery to this and we put the negative terminal to the N side and the positive terminal to the P side, we're going to give enough of a voltage gradient that these electrons will be able to jump that gap that they couldn't before. When they jump that gap, it just opens the floodgate. The electrons all keep going, and we get an electron flow going through this circuit. This is what we call a diode. So a diode in electronics allows electron flow to only occur in one direction, versus if you just put a conductive material like a copper wire, that voltage won't have perfect electron flow in one direction. Those electrons will go up and down, some might even go back. When that happens, it creates heat. With a diode, there still is heat created, but it's minimalized compared to just a wire, which is very important when you're thinking about an electronic device like a computer. So you're able to keep a flow of electrons in one direction. That's going to be very important in an electronic device because how do they work? Circuits of on and off. Remember binary? We're either letting electrons go through the circuit or we're not. So what gets very interesting about this diode is when those electrons are jumping and they go into one of those holes, they're eventually going to land in a hole. When they land in that hole, what's actually occurring is those electrons are getting excited to a level, they're jumping from that material, the atom that they're on, and they're settling into a new atom at that excited state, that excited valence state. It's going to eventually settle down to the resting state. So remember back when I talked about an exciting electron with electromagnetic energy? If nothing happens, it's going to resettle down into its resting state and emit photonic energy as it releases it. Same thing is going to happen in a photodiode, a light emitting diode. So when you're running one of those diodes, as those electrons are hopping, they're emitting electromagnetic energy. All diodes emit electromagnetic energy. Only LED emitting diodes emit visible light. So most diodes emit infrared energy as heat. So in a computer, those diodes are not emitting all kinds of frequencies of light, visible light. They're releasing infrared light and they're creating heat. So this is a way that a semiconductor can actually create light. So frequency of light emitted is proportional to the band gap, which is governed by the doping material. I guess I forgot a note in there, didn't I? So what that means is this depletion zone is going to take a certain voltage to give enough energy for that electron to jump across it. The inherent nature of the doping material is going to govern how much energy is going to be needed for that to jump. That energy is going to be equivalent to the frequency of light it's going to emit. So a common photodiode is gallium phosphide. So they're doping that silicone matrix with gallium and phosphide. When that happens and they run a certain voltage through it, they know they're going to get green light emission. So in the photo lighting industry right now, the LED lights we all have in our houses, it's a combination of a red, green, and a blue diode. They basically figured out how to dope to get those certain frequencies. You can combine that red, green, and blue in proportions to get different colors. So if you equally combine them, you're going to get white light. If you want more of a blue light, you're going to have more of the blue LEDs in there. So we're going to get to why this is important in a little bit within biologic proteins. But there's one more situation that we have to discuss. there's another option of how these semiconductors function. I just ran out of room on my board. So I'm going to quick erase this board, and we're going to get to the next quality of semiconductors. Okay, so the next nature of a semiconductor is that you've got this material, the semiconductor, where we have a depletion zone in it. If this depletion zone is exposed to electromagnetic energy, something really interesting happens. That electromagnetic energy will cause these electrons that are in the hole to reach their most excited state, but actually high enough that that excited state is going to cause those electrons to jump. And instead of jumping and going this direction into the next hole, they do what you wouldn't expect. They actually jump back over to the other side. The holes will migrate to the other side. This is very important because I've been talking about this since we started this video series. What just happened? We just created a charge separation. That's what a solar cell is, a photocell. So photocells are set up to excite those electrons and have them separate and create a charge separation. Now, if you were to hook this up to a battery, you would now have a voltage in that battery. That voltage will be proportional to the electromagnetic energy absorbed into that. But in turn, what we were talking about a minute ago, the quality of the doping material, that will dictate the band gap, that will also dictate the charge separation. So visible light interacting with that in a solar cell is going to create that charge separation. So this is going to be important. So I'm going to review this. Semiconductors have the ability to not only emit light, so convert charge separation, voltage flow into electromagnetic energy. It works the opposite direction. A semiconductor can take electromagnetic energy and convert that into a charge separation. So when you're thinking about what I discussed earlier, the pigment system in the skin, that pigment system in the skin is absorbing solar electromagnetic energy, and the makeup within that melanin pigment is in such a way that those frequencies are going to create that charge separation. They're going to create that charge separation in a very specific way, which is going to come into play on how that charge separation then turns into electromagnetic energy created by biologic tissues. So semiconductors within the pigment are taking in that charge separation, then they're transferring that charge separation into those beta-pleated sheets I was talking about a minute ago. Those beta-pleated sheets are going to be organized in such a way that they create a specific biologic frequency that goes into the system. So I'm going to do another drawing here. I'm going to take a pause, clear this up, and I'm going to set something else up here for you to see. So what I drew up on the board here is a segment of amino acids, and I'm really going to make this basic not to make it complicated. We're going to have 4 amino acids in a sequence. So we've got the amino acid backbone, which is the repeating segment of each amino acid. These guys are all joined together. These residues that come off of each amino acid segment is what makes each amino acid unique. So if you think about it as a semiconductor, we're going to be basic in saying, okay, this guy's got a lot of pi electrons. This guy over here holding on to electrons tight. So you can look at this as free electrons, holes. Now, again, I went extremely simplistic. You have to picture that in these amino acid sequences. These guys are rotating and folding onto each other, and there's a core in here of amino acids. On this other end, there could be another backbone here. When I'm talking about that beta-pleated sheet, it can be so many different interactions. There could be 3-dimensional, not just 2-dimensional interactions of these different residues. So these different residues are going to have different qualities as a voltage is placed across this protein. That voltage can come in many different ways. We talked about a very important molecule early on in this, ATP. What is ATP? It's holding a charge separation, it's stored charge. So if you get an ATP to bind to a location on a protein, that's going to induce a voltage on that protein. So you're going to have more negativity where that ATP is bound versus over here where it's not. Once we cleave that ATP and we create ADP, well, now we might be getting an electron to flow through this circuit. So the interaction of volt separation across these residues is going to exhibit multiple types of semiconductor properties. It can emit electromagnetic energy. So when we're talking about the mitochondria within a cell, that red light is activating that cytochrome to create the ROS. Well, that red light's not something that's coming from a red light therapy machine or the sun. The sun is getting that energy into the system. That system is moving this energy through these semiconductor proteins in a very controlled manner. Within a cell, each cell is producing its own electromagnetic energy, its own frequencies to drive all the different reactions, from a cytochrome to create ROS to an enzyme, just a classic enzyme, that electromagnetic energy can absorb into one of the rings within that enzyme to activate a protein, an amino acid transfer to one of the substrates, or pull an electron, to activate an electron to attach to, so oxidize or reduce that substrate. Or the reverse can happen. We can pull electrons from the substrate, oxidizing the substrate to make a chemical reaction happen. So interactions with electromagnetic energy in this system has its own inherent natures that's going to change how the protein functions or drive the function of the protein. But within protein structures in the cell, there's a biophotonic system. And we're going to discuss that. That's within the microtubule system of every cell. That all the processing of everything occurring within the cell is interacting with the cell through different reactions within these protein cores. So we're going to have a whole topic on that, but I want this to get into your head that electromagnetic energy is interacting with these protein segments through these different aromatic rings, but voltage separation through proteins can emit electromagnetic energy from these different aromatic rings, different semiconductor properties on the protein. So I'm hoping I didn't breeze through that too fast. Very important topic. This is going to be the backbone of this entire biophotonic system. All the interactions of proteins are occurring at the quantum level within the semiconductor nature within the core of these amino acids. And how they're constructed, how they're coded from the DNA and the different segments dictates how these different proteins are going to interact with different frequencies. So I hope that was clear. I will be going over this topic over and over again though, but this understanding these semiconductor properties is going to be vital to understand this system. So thank you.